after a neurotransmitter is released, it can either bind to a receptor, be taken back up by a transporter, or metabolized. In this video, we're going to focus on receptor activation. So let's start by looking at ionotropic receptors, which we can see diagrammed here. Ionotropic receptors are responsible for fast neurotransmission, and they are made of 4 to 5 different subunits, and in the center of those subunits is a channel or a pore through which ions can flow. When a ligand binds to an ionotropic receptor, it opens the channel rapidly, which allows ions to flow in, and positive or negative ions, depending on what's flowing through that particular receptor, will cause changes in the membrane potential. So we get rapid changes in membrane potential, which you might remember are called EPSPs or IPSPs, depending on whether they're excitatory or inhibitory, through the function of ionotropic receptors. Now, we can look at the structure here, and as you can see, this receptor is made of 5 different subunits. So this is a protein, and it's really a complex of smaller subunit proteins, which are bound together in this circular formation. And in the middle of these subunits, it's open, and this forms a channel or a pore through which ions can flow. Because remember, ions are located on the outside and the inside of the cell, but they can't pass through this lipid bilayer, the cell membrane. So the opening of the ion channel can allow positive ions to enter the cell to cause depolarization, or maybe negative ions to enter the cell to cause hyperpolarization. Ionotropic receptors are also called ligand-gated receptors because they open when a ligand, which is a neurotransmitter or maybe a drug, binds to the receptor. And so we'll see that these receptors can exist in closed or open states. If we look here at this figure, we can see the ionotropic receptor in its closed state. The channel is right here. This is a figure as if we've kind of cut the receptor in half, and so we can see the inside of it. And we see the channel here is actually closed. So the ions that might want to pass into the cell can't do that when the channel is in its closed state. And of course, the ionotropic receptor is most of the time in its closed state. And it's only when the neurotransmitter actually binds, and you can see the binding pocket diagrammed here, that we get this conformational change in the shape of the receptor so that the protein changes its shape, and this pore in the middle of the receptor actually opens up, and now it allows the ions to flow. And that's what's going to then be responsible for that rapid synaptic transmission. These ionotropic receptors can have a variety of different properties, which will be determined by the conformation and the properties of the proteins and the subunits that make them up. So they can have different permeability to different ions. Some ionotropic receptors only pass sodium, for example, like the nicotinic acetylcholine receptor. Other ionotropic receptors can pass sodium and calcium, such as the NMDA receptor. And that is important because it determines what's going to happen in the cell once the channel opens. Some ionotropic receptors can pass chloride, and that will obviously produce a hyperpolarization response versus a depolarization response. So all of those properties are very important. Another important property of ionotropic receptors is that they can desensitize. So we're going to see this particularly again with the acetylcholine system, but neurotransmitter, when it binds, doesn't always open the channel. The protein can kind of enter this state of desensitization where the binding is no longer effective until the channel sort of resets. And so that's another important property of these channels that can determine how they affect the function of the cell. Now let's talk about metabotropic receptors. These are sometimes called G-protein coupled receptors because they're coupled to an intracellular signaling cascade. There are many different molecules in the cell that can all serially affect one another to ultimately affect the function of the cell. And the metabotropic receptor is sort of the first messenger. And then it'll cause changes in a second messenger, which goes on to affect that whole signaling cascade. So let's take a look at the structure of a metabotropic receptor. So we can see that metabotropic receptors are actually made of one subunit. With the ionotropic, we saw that they were 4 to 5 subunits coming together. Metabotropic receptors are made of one very long subunit. So all of this is one protein. But it has 7 membrane-spanning domains. And so you see these diagrammed here, that the protein spans the membrane, then it has an intracellular part, it comes back across the membrane, and there's an extracellular piece. These extracellular parts of the protein will be what bind to the neurotransmitter, and then the intracellular parts are the ones that start the signaling cascade. And so we have neurotransmitter binding sites extracellularly, and G protein binding sites intracellularly. When the neurotransmitter binds, again, it's going to cause a conformational change. It will change the shape of the protein, and that'll activate the G protein that's bound intracellularly. So we're not passing ions across the membrane like we did with ionotropic receptors, but we are influencing what's happening inside the cell via something that's happening outside of the cell. So what actually happens when these G proteins are activated? We can see 2 examples of this here. In one case, we could have neurotransmitter binds to the receptor, causes that conformational change, which activates the G protein. The G protein can then go and cause a signaling cascade, which would activate an ion channel. So these are frequently actually potassium channels, and you might have potassium channels that are in the membrane and they're closed, but when the G protein activates this channel, it can open up. So this is a G protein gated ion channel, which would then allow potassium ions, which tend to flow out of the cell, to leave the cell and cause a hyperpolarization. So one mechanism through which metabotropic receptors work is activation of an ion channel, which would then cause a fairly rapid change in membrane potential, although not as fast as with ionotropic receptors. And then a slower signaling cascade might be if the G protein activates an effector enzyme. And so an enzyme then is going to go on and activate another messenger, and we will see now this entire signaling cascade be activated. Second messenger signaling cascades very often activate protein kinases. Kinases phosphorylate proteins, and that phosphorylation will affect the conformation and the function of the protein. So you can see this diagrammed here. The protein kinase will phosphorylate a protein, and this can have many cellular effects. Some examples might be opening of an ion channel, which we've already discussed, changes in gene transcription, changes in neurotransmitter release, or changes in the sensitivity of a receptor to neurotransmitters. So any protein you can imagine that exists in the cell could potentially be altered through this signaling cascade, which begins with the metabotropic receptor. Let's look at some specific examples of intracellular signaling cascades. What we can see diagrammed here is a metabotropic receptor embedded in the membrane. It has the 7 membrane-spanning domains and neurotransmitter bound to the extracellular component of the receptor. On the intracellular side, we see the G protein signaling complex. Now, these G proteins can come in different varieties. GI is for inhibitory, GS for stimulatory, and GQ. GI and GS work on the same second messenger cascade, but they produce opposite effects. So GI, which is inhibitory, inhibits adenylyl cyclase, which is the effector enzyme in this cascade. GS, which is stimulatory, stimulates adenylyl cyclase. So we can know something about a receptor based on whether it's coupled to GI or GS. So these are the G proteins. Adenylyl cyclase is the effector enzyme. And then this enzyme goes on to activate cyclic AMP, which is considered the second messenger. This second messenger then activates a protein kinase, in this case, protein kinase A. And the protein kinase phosphorylates a variety of other proteins, which can then go on to affect many different components of the cell. GQ is another protein, G protein, and it also tends to have an excitatory effect, but it doesn't work through adenylyl cyclase. In this case, the effector enzyme is phospholipase C, which then goes on to influence IP3, DAG, and calcium. These are the second messengers involved in this cascade, and they all influence the protein kinase C.